I'm a master's student at USM's Gulf Coast Research Lab, and I'm actually one of the few people in America that are currently studying marine fungi. Today I'm going to discuss my thesis entitled On the Rocks, the first look at mixed species fungal communities in biofilm on artificial reefs in the Mississippi Sound. Currently, there are over 65 artificial reefs in the inshore Mississippi Sound. These artificial reefs are important to our community, both ecologically and economically. Economically, they are important because they increase fish assemblages and also amplify our oyster habitats. Ecologically, the artificial reefs are important because they protect the shoreline from storm surge. They help to stabilize the sediments, and they can also promote good water quality. Now, currently, our artificial reefs are monitored by only measuring the fish assemblages, and those can really only be measured whenever the artificial reef is mature. However, the reef really can't become mature until the basic trophic requirements have been met. So these microbial biofilm communities can be really important in the last stages of succession, because the most accepted successional model states that later stages of succession are facilitated by the early successional species. Fungi, along with these other microbes, they'll immediately form a complex biofilm on all surfaces that are immersed in seawater. Like I was saying before, these early successional species, which are the biofilm components, they can dictate which organisms will adhere to their certain substrates. Many studies have been done that have shown that marine invertebrate larvae will not settle or metamorphose until they encounter specific conditions that will ensure their growth. Settling larvae will react to the physical, chemical, and biological properties of the substrate, and they can even exhibit specific behaviors whenever they contact a suitable biofilm. The two main objectives for this project are to compare the fungal biofilm diversity both seasonally and between the reef sites. 2 high-profile artificial reefs in the Mississippi Sound were chosen in both the east and the west. Well, one in both the east and the west. Square Handkerchief Key is in the west. It's just off the mouth of Bay St. Louis, and it's composed of crushed concrete bridges. Katrina Key Reef is located in the east, off of the shore of Deer Island, and it's composed of limestone rock. These substrates were taken with 14-foot-long oyster tongs, and then also abiotic factors were measured at each subsite. These samples were consistently taken at 3 subsites within each reef site. The reason that these 3 subsites were chosen was because they make up the area of the reef that is newest, because DMR is constantly replenishing the reefs to keep them from settling or being covered by sediments. Right now I'd like to point out the substrate, especially the picture on the left, which was taken from Handkerchief Reef in January of 2010. Notice there's no oyster growth at all on those rocks. And the substrate in the middle picture was taken from Handkerchief in March of 2011. And as you can tell, there are adult oysters living on the substrate. So clearly the biofilm composition that was here was conducive to oyster settlement and growth. There are 2 different ways that the fungal biofilm community are analyzed. The first way is by morphological culturing methods. Initially, the biofilm is plated on to antibiotic marine agar. It's allowed to incubate until mycelia is observed. And then the unique fungal colonies are plated on PDA, which potato dextrose agar. It has extra nutrients in it that will allow the fungi to sporulate. The only way to identify fungi is by observing their fruiting structures. So even if a species will grow on the agar, you cannot identify it unless it's reproducing. Furthermore, there are lots of fungi, especially from the marine environment, that will not grow or sporulate at all in culture. So to account over 25 genera identified by morphological ID. These genera will span the Ascomycetes, Zygomycetes, and Basidiomycetes. And these have even included a few yeast Basidiomycetes that could possibly be new species. Now, to account for the fungi that will not grow in culture, we use molecular methods. The fungal ITS region of ribosomal DNA is amplified with specific primers. This ITS region was chosen because of its high sequence variability at both the genus and the species level. Now, terminal restriction fragment length polymorphism, or TRFLP, is a process by which you label each ITS fragment with a fluorescent tag at the terminal end. And then you digest that product with HAE3 enzyme. This enzyme will cut the DNA at a specific place between the bases GGCC. These fragments are then run through capillary electrophoresis at an outside facility. These restriction fragments that come back to us in a chromatogram, which are the graphs that are on your right, each peak will represent, it's actually a proxy for the abundance and different species. However, since we cannot be exactly sure if each fragment represents one species or possibly more than that, we call each of these peaks a phylotype. Now, these chromatograms came straight from GeneMapper, but I have overlapped them for each site so that you can see the ones that they have in common. Here in late summer of 2009, there are 5 phylotypes that these sites have in common. One at 55 and then another at 59 base pairs, and then also 179, 441, and 474. In fall of 2009, they also have 5 phylotypes in common. These are also at 55 and 59 base pairs, 80, 180, and 298 base pairs. In the winter of 2010, these sites only had 4 phylotypes in common. Again, at 55 base pairs, 74, 131, and 298. Now, if you notice, in all 3 of these sites, there is a phylotype located at the 55 base pair mark. Now, this could be really important because it could show us that this could be a key fungal species within biofilm community processes. This chart shows the number of unique phylotypes at each artificial reef. If you'll notice, in late summer of 2009, there are a less number of phylotypes than later on. This is probably due to the fact that the reefs were put out very close to the sampling period, so this stage is probably the earliest successional stage. However, as you can see, the trend is slightly increasing since then, so we would expect to see increased numbers of phylotypes with time. This graph shows the Shannon Index of Diversity for each subsite at each date. The diversity greatly varies, and really it doesn't show any seasonal correlation, which we had hoped that it might. But overall, you can tell that the sites at Handkerchief have a slightly higher diversity than the sites at Katrina Reef. That could also play a part in the oyster settlement and recruitment that I discussed with you from the previous slide. Some preliminary conclusions that we can make from the data thus far is that there's no distinct seasonal differences in community structure. Overall diversity for both reefs is slowly increasing. The overlapping phylotypes are most likely members of the resident fungal community, while the phylotypes that occur less frequently are probably transient members. Lastly, there will be further statistical analysis performed using a repeated measures design once more data becomes available. I'd like to thank the Mississippi Department of Marine Resources and my committee, as well as the CAP grant for funding. Does anyone have any questions?
How long does it typically take for the incubation period for the reproductive structures to show up?
Initially, the colonies will start growing within 3 to 4 days, and then depending on the species, they could sporulate immediately, or it could take them weeks or possibly months. So it depends. Yes?
Do they produce mycelium while they're growing in the biofilm, or do they stay in single cell study?
There are filamentous fungi that will live filamentously in the biofilm, and that actually helps to structure the biofilm because well, not lots, but a little bit of research has been done on that, and actually we found that bacteria will form biofilm on top of living mycelia. So it's a very new field, fungal biofilms is, so it's really exciting to be able to see exactly how they're growing and how they're interacting with the other members of the biofilm community.
I was just curious, is this biofilm, or is it important for the settlement of any certain species that you know of? Or is it...
Not that I know of, simply because I'm looking at everything from the mycology point of view. But if I were to, work with the benthic department, then yeah, I'm sure they could tell me of species that, are very particular about what is in their settling community. Anything else?